Hello dear viewers, welcome back again in Glittering Biology with your Bio Guru Alok. Here, once again I am along with you with the same chapter of Unit 5, that is Digestion and Absorption. And till the last chapter, I told you about the different parts of the alimentary canal along with its function. Now today we will just talk about the different types of glands in short. Later on we will just talk about the digestion mechanism. So let's start it with the digestive glands. There are the several glands which are involved in the process of digestion. These include salivary glands, gastric glands, liver, pancreas, and the intestinal glands. If we will just talk about the salivary glands, then these are the special glands which are just providing a special enzymes for the digestion of food material. They just lubricate the oral cavity, lubricating the food and dissolving chemicals that can stimulate the taste buds. Most important is the salivary amylase enzyme that begins starch digestion in mouth. Second enzyme is the lingual lipase enzyme that is activated by the stomach acid and digests lipid after the food is swallowed. Mucin, uh, isogyne, these are the certain other enzymes which are just produced, which are present in the saliva. Immunoglobulin A, IgA, an antibody. It is also present along with the saliva, which inhibits the bacterial growth. After that, there is the second main glands are just related, called as the gastric glands. There are two or three tubular gastric glands open into the bottom of each gastric pit. That are the three parts what we have already discussed into the stomach, cardiac, fundic, and the pyloric. So what they are just doing, they are gastric glands contains different types of cells which are producing a specific hormone or enzyme. In that we can just call we can just keep the mucus neck cells which secrete mucus. In the second stage, the parietal cells that is found mostly in the upper half of the gland secreting hydrochloric acid and the intrinsic factors. Hydrochloric acid, it is very important part of our digestion. It makes the food, it makes the medium acidic for its digestion. Enterochromaffin cells are also present over there. They secrete histamine, stimulates hydrochloric acid, and beside that there is the specific D cells which secrete a special hormone that is somatostatin. G cell which secretes the hormone gastrin. These are the specific cells which are present inside the gastric region of uh, alimentary canal. Next it is the gland which is the liver, very important, and I told several times it is the largest gland located above stomach under the diaphragm. It has near about weight 1.4 kg, and it is liver is also known as the heaper, and for that particular we are just using the hepatic artery, hepatic vein. Here no digestive enzymes are produced by the liver. It is just covered by the liver capsule and the thin layer of connective tissue separating the lobules, that is the Glison's capsule, clearing of poisonous substances from the blood, resisting infections by the producing immune factors and removing bacteria from the bloodstream through the special cells that is called as the Kupffer cells. That is also a very important function of that one. Next, it is the pancreas, a very important exo as well as the endocrine gland of the body. Uh, it is the endothermal in origin and called as the mixed gland or the heterocrine gland. Exocrine part contains the pancreatic acini embedded in the connective tissue. Each acinus is made up of the pyramidal glandular cells, pancreatic cells around a central lumen. Secreting cells of acini releases a mixture of digestive enzyme called as the pancreatic juice, and that one is just delivered through the special duct. That one is called as the pancreatic duct. Then the accessory pancreatic duct, the duct of Santorini, may also lead from the pancreas to duodenum. Its endocrine part contains islet of Langerhans. They contains four types of cells: alpha cells, beta cells, delta cells, and F cells. That is also called as the pancreatic polypeptide cells, PP cells. Functions are given on the same page, you can just see over there. Now next topic is the physiology of digestion. We need to talk about this one, that ingestion. The term ingestion is taking food into the buccal cavity through the mouth. In buccal cavity the food undergoes following changes: its pieces has been formed over there, salivary glands are pouring saliva on that one, enzyme salivary amylase starts digestion of carbohydrates, and taste buds are present on the tongue also try to capture the taste of that food material. Swallowing or the deglutition, that one is the term for the bolus whatever is formed in the mouth, they are just released towards the food pipe. Oral cavity is involved in this one, and they are that bolus are just passing through the pharynx towards the esophagus. There are three steps are involved: voluntary stage, pharyngeal stage, and the esophageal stage. So during swallowing, glottis remain closed and breathing stops for a moment. It is a flap-like cover, which one is just covering the food pipe as well as the windpipe according to the requirement. So whenever we are just swallowing something, at that time epiglottis just cover the windpipe for a while. Now next one is the digestion. We are just going to talk about the digestion of food. Digestion is defined as a conversion of non-diffusible food elements into the diffusible constituents. You can just say that it is a conversion of the large molecules into the small ones. There are the two types of digestion observed in the organism. One is the mechanical as well as the chemical. Whenever we will just talk about the mechanical digestion, at that time we will just discuss it under the mastication, liquefaction, and swallowing, means deglutition, whatever we have already discussed. Mastication, what it is, chewing of the food material is just considered as a mastication, and liquefaction of the food, while we are just mixing the saliva or the different types of other liquids during the digestion. Then deglutition, which I already discussed, that one is divided into the three phases. The first phase is the pharyngeal stage. What happens over there? Esophageal stage is the last one. The swallowing center is located in the medulla oblongata and lower pons varolai. Just note down that one. Means what is when we are just completed the mastication and the liquefaction of the food material, at what time it is ready to be swallowed, that one is just present along with the medulla oblongata and lower pons varolai. That one is one word answer for you all. Then there is one more term is for you, that one is the peristalsis. How that bolus whatever is formed, that one is moves into the alimentary canal. That is a special mechanism in the human beings or the that vertebrates. The wave of relaxation followed by one of contraction. That wave is just pushing the food propulsive manner downward towards the stomach in the esophagus. This is called as the peristalsis. Whenever we'll just talk about the chemical digestion, then chemical digestion is required the different types of enzymes for digestion. It includes the enzymatic actions which are already the proteinaceous nature. They are hydrolytic, means the enzymes are hydrolytic as they catalyze hydrolysis of the nutrients. During this entire process, the small amount of the energy is released as a heat. Four main types of digestive enzymes are there: carbohydrates, carbohydrases, proteinases, lipases, and the nucleases. In the carbohydrases, what we will just contain there? Carbohydrases includes amylases, which breaks the polysaccharides to disaccharides, and disaccharidases are just supporting the maltase, sucrase, and the lactase. These are the enzymes which are just studied under the carbohydrases. Then proteinases can be endopeptidase or exopeptidase. Next is the lipase, fat digestive enzyme. Proteinase, what it is? They are just protein digesting enzymes. Protein को digest करने वाले enzymes हैं. And lipase are fat digestive enzymes. What they are? They are converting the fat into the fatty acid or glycerol. Then the last is the nucleases. Nucleases are the deoxyribonuclease or ribonuclease. Generally, they are used. They split DNA and RNA into the specific form. Nucleotidase split nucleotide into the nucleoside and inorganic phosphate. Nucleosidase split nucleosides into the nitrogenous base and the pentose sugar. Means they are just making their simpler form over there. Then whenever we will just talk about the digestion in mouth cavity, earlier I told you that mastication already has been done. Whenever we will just talk about the chemical digestion over there, then the function of the salivary amylase or the ptyalin is observed over there on the starch, which convert it into the maltose, isomaltose, and alpha dextrins. This is the product what we are just getting from the starch by the activity of the salivary amylase. Here it is also given lysozyme, whatever is present in the saliva acts as an antibacterial enzyme. Earlier you have just studied about the lysozyme in the sense organs, especially in eye. Digestion of the proteins and fat do not occur in the buccal cavity because saliva does not contain any proteolytic enzyme and lipase. So in the mouth cavity you have to just consider the starch along with the ptyalin, whose pH is near about 6.8, and it is converting the starch into the maltose or isomaltose along with the alpha dextrins. Next is the digestion in stomach. Now in the esophagus there is no digestion, only the bolus are just thrown in towards the that stomach by the special peristaltic movement. Here in the stomach, the gastric juice has been secreted, whose pH is near about 1 to 3.5. Means earlier as I told you that HCl, hydrochloric acid, has been produced there, so that one is making the medium more acidic. Protein digestion starts in the stomach, chief site for the protein digestion. Food mixed with the gastric juice in the stomach, and it is called as the chyme. Then what happens? Chemical digestion. Just have a look of that one. Gastric juice contains the mucus, hydrochloric acid, and two inactive proteases, that is the pepsinogen and the prorenin. Pepsinogen and prorenin, what is the main function? They are inactive, means they only work for a while to activate the process. Okay, so these enzymes are performing a special function like mucase. Mucase, what it is? Secreted by the goblet cells, protect stomach wall from the hydrochloric acid or the hydrochloric acid activity. Here hydrochloric acid is playing a very important role for the digestion. It, what is the main function of that one? It softens the food and disinfect by killing bacteria. Action of salivary amylase stops over there due to the very low pH. Activates pepsinogen to pepsin and prorenin to renin. Means here the digestion of proteins has been started with the activity of these two inactive enzymes. Maintains appropriate pH, that is near about 1.4 1.8. That is the average what I told you, that from 1 to 3.5. Whenever you will just get the average of that one, it is mentioned over here. The control functioning of the Castel intrinsic factor is just managed by the hydrochloric acid, and opening and closing of the pyloric valve is also managed by the hydrochloric acid. Gastric juice does not contain any carbohydrate hydrolyzing enzyme. Means here in the gastric juice, carbohydrate hydrolyzing enzyme is not available. HCl destroys the activity of ptyalin. There is no digestion of carbohydrates in the stomach. Underline that one. Earlier in the mouth cavity, carbohydrate starts digestion itself, but in the stomach, where there is a HCl is present, it will just inhibit the activity of the ptyalin. So there is no carbohydrate digestion observed into the that in this region. Now look the digestion of the proteins, how it is taking place in the acidic medium. What it is given? Proteins are just digested, which starts from the activity of the pepsinogen. Pepsinogen, whenever the HCl comes in the contact of pepsinogen, then it forms a special content, that is the pepsin. What food we are just having in our stomach, if that one is contain the proteinaceous substances, then pepsin, whose pH is only 2, it starts the breakdown of that proteins into the proteoses and the peptones. These are the peptides. Later on, pepsin, what is the main function of the pepsin? It helps in the coagulation of milk. Coagulation of milk is just a process in which the milk protein part has been just detached from the other substances. It hydrolyzes soluble milk protein casein. Underline that one. In the milk, whatever is protein is present, that is known as the casein into the para-casein. So para-casein is the output whenever the pepsin act on the casein, and it will be just form another product, that one is called as the whey protein. Para-casein is precipitated as calcium paracaseinate to form solid curd. This is called as the coagulation of milk. Ever you have seen whenever the small babies are just drinking the normal milk and suddenly they split milk or they vomit milk, that milk is in the different form. So that one is due to whenever that milk is reaching up to the stomach part of the body, there this pepsin makes that coagulation of milk. Now the next one is the prorenin. It is not present in the adults. In infants and the children, active form of renin Its active form of renin catalyzes and hydrolyzes of casein of milk into the para-casein, causing milk to curd. So if anyone asks you what is the function or who hydrolyze the casein, then you must write there renin. Actually, casein converted into the para-casein and whey proteins with the help of renin. This renin is not present along with the adults, found only into the kids and the children. Para-casein and calcium, whenever mixed together, then they are formed the calcium para-caseinate, and calcium para-caseinate again comes in the contact of pepsin, which makes it into the peptones. So here renin is also called as the chymosin, not to be confused with the renin, another word which renin, another enzyme which one is produced by the kidney. Here the spelling of renin is RENNIN. It is secreted by the secreted along with the stomach, while whenever we will just talk about the RENIN, then it is just secreted by the kidney. Now digestion of fats we will discuss in the next lecture. Just read that much part or read the entire digestion with your own self. We will discuss through the lecture in the next video. Thank you everyone.